Um, in this section, there are two speakers, Wasimuddin and Rahul. Both of them will be presumably talk on fast algorithms for, uh, you know, gaining insights from biological data. And we'll start with Rahul. And Rahul Siddharthan is joining from IMSC Chennai online. Um, and over to you, Rahul. You can start.
Okay, thank you very much, Rebecca, and thank you to the organizers for inviting me and accommodating me uh, virtually, as it were. So just to begin, slightly off topic, I don't know how many of you know the name of Isadora Duncan. She was down in the early part of the 20th century, and sometime in 1927, she was riding a car with a scarf wrapped around her neck. In those days, cars had spokes, and the scarf got tangled in the wheels of the car, and basically she died. And something similar happened to me, except it wasn't a scarf, it was straps from a bag, and it was a bicycle, so I'm still here and able to talk to you. So just public safety warning: if you're on any two-wheeler or any vehicle with spoked wheels and you have anything dangling, just drag it up. So with that, and as for this talk, I wasn't actually sure what to talk about in terms of fast algorithms. So I hope this kind of fits. It's more of a basic biology problem, and it's something we haven't published yet. We're working on. So I'm guessing you've heard about transcription factor binding sites from many speakers so far. So I won't go deeply into that. But roughly speaking, these are proteins that bind in. Okay, to begin with, this is work that is in collaboration with Pavitra here. And transcription factors are DNA binding proteins that tend to recognize short patterns in DNA. And those patterns are often represented as sequence logos here, but they really come from something called a position weight matrix or a position specific scoring matrix, which gives you a position specific probability of every nucleotide. So if this is a CTCF if you have a CTCF binding site, this kind of pattern tells you that with 100% probability, you'll get a C here, you'll get a G here. With slightly less than 100% probability, you get a C here, and so on. Whereas at certain other sites, it's really a toss of what nucleotide you might see. So this is a position-specific weight matrix, which is commonly used. Now, what we are asking here is a sort of related question. Supposing you consider a site that is it's evolving because it's a locus in your genome, but none of us have exactly the same genome. We're all evolving, and we've all evolved from some primates with whom we have a common ancestor as a chimp, and even further back, the vertebrates and so on. So how are these sites evolving? Are there sort of selective pressures on them? And that's the kind of question that we are analyzing here. So the rough plan of the talk is going to be how does DNA evolve, and a rough overview of various models of molecular evolution. And then we'll talk about this particular algorithm F81, which is it was one of the earliest algorithms for its kind. We'll talk about it, and it has some nice properties that are useful for us. And then how do we apply this to this problem of binding site evolution? I should mention, so who else has looked at this problem? We couldn't really find anything exactly the same, but this paper from 2003 does look at kind of the same thing in yeast. And in those days, there was much less data available. There are a couple of differences from what we do. They assume that, so they're using a model called the Halpern-Bruno model, which combines mutation with selection, and they're looking at selective pressures. But they assume that the stationary distribution at any position is given by the weight vector. Now, we are basically sort of combining the selection and stationary distribution questions. We're using an evolutionary model with non-uniform stationary distributions. I will describe what all that means. And inferring at each position, for example, in this, what is the equilibrium position here? So this logo came from a collection of a few tens of sites in human, all of which have G at this position. But now, if you look at a population that is evolving over a long time, will you always find a G here, or will you find some other distribution? So that's the question. Okay, so let's talk about DNA evolution. How does DNA evolve? It evolves because basically it can cause changes when it replicates. It can also change because of mutagens, whether it's cosmic rays. And these changes can be of various kinds. You can have single nucleotide substitutions, which is what people normally think of when they think of mutation. You can have insertions and deletions. You can have segmental duplications. You can have recombinations. You can have whole chromosome duplications, whole genome duplications. There's a huge arsenal of ways in which DNA evolves, and it's evolved all the way from one last universal common ancestor to all the species you see today. But for the purposes of phylogenetics, most models out there even today consider only single nucleotide mutation. So the idea is basically you start with a DNA sequence. So if you look at the history of phylogenetics, earlier it was based on morphology before DNA sequences became available. So they looked at traits, they compared traits and which ones are similar in terms of traits, which could be phenotypic traits. But now we look at DNA. And to make life simple, usually all insertions, deletions are thrown out and you only look at the conserved sequence. There are more sophisticated models, but we won't go with those. This is a basic toy example of how it would go. Supposing these were on the left were the 5 sequences that were given to you, you might ask which sequences are most similar in terms of traits. And now the traits are just the nucleotides at the specific positions. And just by a comparison, you might find that human and chimp are extremely similar. They differ only in these positions, 2 positions. Similarly, mouse and rat look very similar. They basically differ. This is a cooked up example. Don't look at a real genome for these. But based on this, there are algorithms that will let you construct a tree like this, which suggests that human and chimp came from some ancestor, mouse and rat came from some ancestor. These in turn came from some ancestor, and cow is a little bit further away. And instead of using a sequence of just 10, 15 nucleotides, you would use the genome as you could, but you would look at parts of the genome evolving not under strong selective pressure and so on. So it is a very complicated field, but let's assume that we're talking about neutral evolution at the moment. So you can, one concept that we have to understand is, given 2 sequences, a distance between the sequences. And the crude measure would be the number of mismatches, which is what I did in the last slide, 2 mismatches between human and mouse, human and chimp, and so on. But what you really want to get at is the evolutionary time when they diverged, and you can get, and the way this is done is you assume an instantaneous rate matrix. In my notation, it's R beta alpha, which indicates the rate of instantaneous rate at which alpha mutates to beta. And if you are given this matrix, then can you calculate a transition matrix, basically a probability matrix of seeing beta at a later time t plus t, given at alpha at time t. So these 2 matrices are not the same. This is instantaneous, and this could describe multiple mutations, like alpha to go to some other gamma, could go to delta, which then go to beta. So you could start with an A and end back with an A, but there could have been many mutations in between. If you have a matrix like this, you can actually estimate the joint likelihood of 2 sequences given this matrix and given the evolutionary distance between them. And if you want to estimate the distance, then you maximize the likelihood for 2 sequences. And we'll get to larger sets of sequences shortly. But that's the basic structure of phylogenetics. So how does this work? How do you calculate the T from the R? So sorry for a slightly complicated slide, though some of you would know all this very well, but it's probably not a mainstream topic in this meeting, so I thought I'll go through it. So supposing you have this matrix R, then the number of nucleotides at a given locus is, with a population size n, is n sub alpha. So the fraction is this divided by n. And then you can ask, how does this n sub alpha change with time? It changes because the n sub beta changes because beta is getting mutated into something else at the rate given by this, but also other things are getting mutated into beta at a rate given by this. And both of these look like matrix multiplications except for this alpha not equal to beta. So you can basically consider this piece, which is a sum only over alpha, not beta, and we simply define the diagonal elements of R as this. So then you can write this as a standard rate equation, and it has the solution that looks like this. And if you want to now ask the probability question that if the initial nucleotide was alpha, just take the population fraction to be alpha, and then this is the beta alpha element of this exponential matrix. So that's the basic idea here. The transition matrix is the exponential of the instantaneous rate matrix, whose diagonal elements are defined as minus of the sum of the columns. And different choices of R determine different transition matrices T. And once you have a matrix, you can do all these likelihood calculations. So how do you choose a matrix R? So okay, so before that, there are some properties that this T matrix satisfies. One is if it is time reversible, which it should be, it satisfies a property called detail balance, which essentially is, okay, so this is called the stationary distribution, which is what we were looking at. Like at any position, what is the distribution of these four nucleotides? For example, in the middle position of CTCF, this is almost 1 and the rest are almost 0, but for most of the genome, you'll find 0.3 for A and T, 0.2 for C and G roughly. So whatever this pi is, your T matrix must satisfy this property at that locus, that the sum, not the sum, there's no sum here, the transition from beta to alpha times the weight of beta should be transition of alpha to beta times the stationary weight of pi alpha. By definition, it doesn't evolve. So it must, this is the condition for stationarity under T. It also has this nice multiplicative property. Actually, I should have written these factors backwards, but if you do that, you know, this factor next to this factor next, it looks like a matrix multiplication again. If you have a nucleotide that gamma, ancestral nucleotide gamma that evolves to beta, and then beta evolves to alpha, and you don't know what beta is, so you sum over all beta. This is equal to the probability of gamma evolving to alpha over the combined time T1 plus T2. And that's important. And that gives rise to something that Felsenstein called the pulley principle, which is that you can't determine the root of a rooted tree uniquely without external information like outgroups. For example, if you have a tree like this, let's say this is your tree, this root is not uniquely, these two branch lengths are not uniquely determined. This call this T1 and T2. Only the sum T1 plus T2 is determined, and you can prove that by combining the previous 2 points. And so you can basically slide this root around. You can even slide it until it joins this guy. You can even slide the root here. All of them are the same tree. They will give you the same likelihoods. You cannot say where the root is without adding an outgroup, which means another species which is far away from all of these. And then you can't determine the root of that combined tree. I'm sorry if I'm belaboring the point for those of you who are very familiar with all this. So how do we choose R? R, there are various choices historically going back to 1969, which is the simplest choice possible. All mutation rates are equal, all of diagonal rates, and the diagonal ones have to be such that the columns sum to 0, so it is this. This was regarded as unrealistic, and this is for neutral evolution, basically. It was regarded as unrealistic because we actually know that transitions, which is A to G or G to A or C to T or T to C, actually happen much more commonly for biochemistry reasons. So it's in practice, you see something like 5 to 10 times higher rate for transitions compared to transversions. So Kimura slightly generalized this model to have different transition and transversion rates, mu for transitions and nu for transversions, and the off diagonal is this. But both of these models the matrices are symmetric, and for any symmetric matrix, you can show that the stationary pi is equal 0.25 each, which is not representative even of a real genome with neutral sites. You know that A and T are more common, C and G are less common, and it is definitely not representative in sites within a TFBS where there could be quite a large skew. So the simplest model that allows non-uniform rates was given by Felsenstein. This is a classic paper where he gave this model, and he also gave an algorithm to calculate likelihoods, which is still used today, which we will describe. So this rate matrix here basically says that the rate of mutating into any nucleotide is just the equilibrium property of that nucleotide, which looks kind of odd. And the diagonal matrices diagonal elements are chosen so that the sum of the columns is 0. Since the pi sums to 1, the sum is 0 for each of them. And if you calculate T by taking e to the R, you find that this R has this nice property that it's square apart from mu is just minus r, and so you can actually calculate the exponential, and what you end up getting is this. So this t also has this nice property that if you think of the mutation rate mu, e to the minus mu t is the probability that in time t there has been no mutation at all. If there has been no mutation, then the probability of seeing alpha is just delta alpha. Either it's the same or the probability is 0. Or there could have been a mutation. There could have been more than one mutation. This is the probability of one or more mutations. And then it says that the probability of seeing alpha is just the background probability pi alpha. So this rather trivially gives you whatever pi vector, stationary vector you want. In conversely, if we do not know what pi is, but we do have the data, we can infer the pi from this, which is what we'll be doing. And we use units with mu equal to 1. That is, you just choose a unit of time such that the mutation rate is 1. And you define a new variable q, which we, in some earlier work, we call the proximity. And in terms of this, you can write this as this. Q is just, in this case, the probability of conservation. And 1 minus q is the probability that there has been at least one mutation. So this turns out to be a convenient notation for us. This also does not distinguish between transitions and transversions. So Felsenstein himself generalized it a little in '84, but the more widely used version is HKY from 1985, which generalized it rather simply that they just put a kappa here in the off-diagonal elements. Sorry, not the off, in the transition elements. This is A to G, and this is, so the ordering here is ACGT. So this and this are transitions, this and this are transitions, the rest are transversions. And as before, this stars are minus of the sum of these. And we can choose time units as with mu is equal to 1 as before, and we can define the same variable q, but it's no longer simply the probability of conservation. If you want to calculate the transition probabilities for HKY, it's a big mess, and we'll only the transversion for cases other than well, the transversion generally has this form. Transitions look more complicated and identities look more complicated. But in the standard language in this literature, what we call Q is basically what other people sometimes call E2 for the HKY. So we're going with that. But I won't talk about HKY much. So let's move on to how to calculate likelihoods before getting to the results. So the likelihood at a particular locus X of seeing some set of nucleotides at those leaves. So an example is here. Let's say you're looking at position 3 of the CTCF binding site, for example, and in human you see A, in chimp you see C, and in mouse you see T. Those are what you see. And the distances between human and chimp from their common ancestor are Q1 and Q2, and this is Q3 and this is Q4. And alpha and beta are unknown nucleotides at these loci. So what is the likelihood given this tree, including given these branch length skews, of seeing A, C, and T? That is given by this expression. Let's go over it backwards. The probability of seeing an A, if this was beta, is T beta, T A beta, which is this guy. Similarly, the probability of seeing this is this guy. And you do not know what beta is, but you do know that beta came from this guy alpha. So you put in here and you sum over alpha, sum over beta. Now alpha also gave rise to this, so you have it here, and you don't know what alpha is, so you put pi alpha, which is the stationary distribution for alpha, and you sum over that. So that's the likelihood. And given any tree, you can kind of inspect it and write it down, but it's kind of laborious to do that. So the standard algorithm is again given by Felsenstein in the same paper. It's basically, I might have to go back and forth between these slides, or maybe I'll just skip this, but essentially you define this variable, likelihood sub I of alpha is the likelihood of leaves below node I, given that the nucleotide at node I is alpha. And for example, likelihood at this, let's number these nodes as 4. This is 1, 2, 3, 4, and 5. The root node is 5. So the likelihood sub 4 of A is the likelihood of seeing A and C here if this nucleotide were A. And going back here, for the leaf nodes, you can set it as simply delta functions because it is what it is, Kronecker deltas. And otherwise, you can calculate it from the leaves recursively. So for any node alpha, it is the probability of transitioning to its children. This is for a binary tree. And so you can terminate it at the root node and calculate the likelihood of the full tree. So it's a very elegant algorithm. It's called Felsenstein's pruning algorithm. And if you implement it naively, you'll find that it blows up. You need to cache the calculations at every stage. It has a complexity that looks, it's linear in the number of species, linear in the number of sites, and quadratic in the size of the alphabet, but that's not a big issue because our alphabet size is 4. So one of the things that I wanted to mention before getting to results is that we have a nice way to calculate it faster for the F81 algorithm. And that's the only reason that I might be in this accelerating biology section. I don't yet know how useful this really is because the F81 model is not terribly widely used, but it has this nice property that either it's mutated or it has gone back to the background. So if you apply this to, suppose you define a star node as a node like this, where all the leaves are, all the children are leaves. So let's take this node, for example. You consider this node and you apply this formula to this node. So this came from here. So with the probability Q5, it has not mutated. So you could write it as Q5 times this tree, where these two leaves are now attached to the root. And with the probability 1 minus Q5, it has renormalized to this pi value. So now you sum over it, but there is no alpha here. It is a completely disconnected tree. So you can reduce this tree to a sum of 2 trees, and then you can keep doing that. So for this guy, you can now pick up this node and do the same thing. For this guy, you can actually reduce this. So this is already in the right form. So basically, it turns out that any tree can be reduced to a sum of products of star trees. And why is that important? Because for a star tree like this one, or like a tree like this, you can calculate the likelihood without recursion just by product over all leaves and the sum of the ancestor. So it's kind of a trivial calculation. So this is the thing. I think I'll skip over the details of this. But essentially, okay, I wrote that the complexity is order n squared L sigma. Actually, I'm not completely sure of n squared, but this works only when n is relatively small, like less than about 10 to 15. But when, and I should also say that I have implementations both for this and for Felsenstein, but neither of them is super optimized. So this is basically, what I've been calculating on a workstation for the data that I will show you. But this is some benchmarking with synthetic data. So just generating trees with various numbers of leaves, that is various numbers of species. In our case, we'll be looking at this region, which is 7 species. But up until and about 13 species, well, up until about 12, our algorithm is significantly faster. And in this region, it's something like 40 to 100 times faster in real terms. And as I said, neither is fully optimized. But Felsenstein, I think, is about as optimal as it can really get. Whereas our algorithm can be further optimized by not representing it as trees at all. Trees are slow data structures. I haven't gone into that yet. If you zoom in on this region and blow up the fastness by 40 so that you can compare, in this region here, it is our algorithm for about 17 is about 40 times as fast. And here it's probably like 100 to 200 as fast for smaller trees. What about scaling in terms of the number of the length of the sequence, which is perhaps the more important thing? You're aligning. Okay, I think I got the wrong image here. Sorry. So it turns out that both algorithms scale more or less linearly from like 1,000 to 100,000 sequences. There's not much difference, but we are again faster by basically this factor. Okay, with that, the last thing I want to mention is you can actually infer ancestral nucleotides using a sort of Bayesian formula. Like what's the likelihood of the ancestor being alpha? It is proportional to the likelihood of the leaves given that ancestor times the prior for the ancestor, which you could take to be the stationary vector for that. or the background probability, not the stationary vector at the weight matrix. But this is basically what we do. And then we can ask a bunch of questions about how TF transcription factors evolve. So which is to say, Pavitra obtained these from the GTRD database of ChIP-seq data and found motif instances within these using PWMs of Jasper and orthologous instances using leftovers. So at the end of it, you have for each transcription factor, you might have something like 50,000 sites in human for CTCF. But if you want to be sure there are orthologous instances in all of these, you might have something like 15,000. And in the range of 1,000 to 5,000 for most of the other factors we looked at. Okay, so most of my talk is methods because the results are still coming. It's a work in progress, but here are some. So the basic thing is, so the thing above is the CTCF position weight matrix, and we've included a lot of flanking sequence for reasons that I won't get into now, but essentially there are some interesting things there. The first observation is that this is the weight matrix from human sites. This is the stationary vectors per position. That is the stationary distribution you would expect it to evolve to, and it more or less faithfully reflects the weight matrix, but it's less informative. For example, in human, where there's always a G, here you find some proportion of A that it might involve to, which is maybe not surprising because that's a transition which is common. For this C, you have C and you have A, G, or T. Here, the order of things below is a bit different, but that's not really significant. More or less, it's the same. Now, you could ask, and this is something that the Lafis in the audience posed to me 10 years ago, and we've been talking about it since then, that if you look at other species, you usually see a G at this position, but in vertebrates, you often see an A. And why is this A, why does this mutation from G to A happen? It could be because, CG could be it's the dinucleotide does tend to deaminate into an A on its sister strand, not on the strand. But we also see that the next nucleotide is sometimes not a C, but also a G. So supposing your ancestral nucleotide was an A, or if the ancestral nucleotide was a G, you see a difference in the motif. So this is a position-specific stationary vector where 50% of the ancestors had a G at this position. In fact, the stationary vector gives you much greater weight of a G here. This is a stationary vector where 50% had an A. And you do see some interesting effects. If the ancestor had an A, then you're much more strongly selecting for a C at the previous position. And you're slightly preferring a G at the next position. In this case, there isn't very much else you can say in terms of differences. Maybe you could say that this G is slightly less required and so on. You'll see there are quite a few other things to say about CTCF that hopefully we will explore in future. But looking at some other factors, this is REST, which again kind of mirrors it. But one interesting thing to me at least is that though overall the stationary vector is less informative, that means the bias is less strong towards the stationary distribution than within the species. For this position, the T here, it seems to be actually stronger than within the species. So again, we could ask the same question, what if this is a T or if it is a C? So if you have a T at this position, you tend to also prefer a T at the previous position. And so this is a kind of a dimeric motif. If you have a T, then the entire first half of the dimer tends to be more strongly selected for. And whereas this couple of C's over here tend to be more weakly selected for. If you had a C here, it's as if you've got a C and maybe you can get away with weaker signal here, and the rest of it kind of looks the same, except that you see a little bit of more GC-rich background if you look here. If you zoomed in on this, you'll see that most of these are C's and G's. So maybe this C is preferred at promoters, that's possible. But if your ancestor nucleotide is a C, then the stationary vector downweighs this. If it is a T, then it upweighs this and upweighs a T here. And we have similar observations. And so for YY1, we looked at, I think, this position where, yeah, this guy, you could consider all four possibilities. It tends to be C, but if you look closely, you see G, A, or T, and this is a case where not a reasonable amount of sequence data. So let's try picking all four possibilities, A, C, G, or T in this position, and you find that if you have a G here, then this is more strongly selected for, and also this is more strongly selected for, and it seems like there's a more GC-rich background in general. If you have an A here, then you see this kind of secondary motif cropping up. It's not very strong. So these are all sort of observations. I don't know what the significance might be, but basic. And yeah, finally, I mentioned HK585. So is it give you the same results? And this is the PSSP with these two. It turns out HK585 is mostly the same, including the ordering of the weights. But these three guys, which are strong in the original motif, are also a bit stronger in HK585 compared to PSSV, compared to F81. And that might be because if you look at the second position, most likely thing here, it's a T here, an A here, and A here. All three are transitions, and it might be because HKY has this greater transition transversion ratio. So to compensate for that, you need your P vector also to be larger here. Whether this is important in real life, we don't know, but these results are like 50 to 100 times slower. Okay, so thanks for listening to this work in progress. Here's a quick summary of what we have. What's the question that we're asking is, can we ask what is the stationary distribution at an individual locus for transcription factor binding sites? And this tells us something about how the sites evolve. And we have this fast algorithm for F81. And we find that more or less the stationary distribution pattern mirrors the within species position weight matrix. And we can look at ancestral nucleotides and compensator mutations, and maybe this is a useful framework for this kind of question going forward. And because this is work in progress and we have like 9 or 10 minutes left, I'd really like to hear your feedback, or if any of you tells me that this is seriously flawed work, I'd like to hear that also. Thank you very much.
Thanks, Rahul, for this wonderful talk.
Hi, Rahul. Am I audible?
Hi, Chirag.
Yeah.
You're audible and you're half visible.
Okay. So
Yeah.
I guess my question is, in the fast algorithm that you showed, the asymptotic complexity is the benefit coming in the sigma factor, or?
No, it is actually coming from all the pre-factors. Slightly factor 4 from the sigma factor, but it's really because if you look at the Felsenstein likelihood, it's basically a recursive algorithm, and you have to do it at each. So you're doing this for an aligned sequence. You're doing it for each position. You are recursing a tree, and because recursion takes stack space and all those things that, and also the data is fragmented in a computer memory, and basically I think the three factors come from that. And for our algorithm also, as I said, we're using a tree, and I suspect it can be made much faster because for a star topology, you basically have only a bunch of leaves. So you don't need to treat it as a tree, you can treat it as an array. And I think that would make it much faster. I haven't done that yet. But it's all three factors.
Okay, so what is the time complexity? What was the?
It is this what I wrote.
Okay.
Except I'm not sure about the n, because I think so one factor is when as you expand this list of lists, how does it scale with the number of leaves? Naively, I thought it scales as n. That is, if you have n leaves, you'll have order n elements in this list, each of which will have a total of n leaves in them. For example, whether you have this term, this had 4 leaves originally, this has 4 leaves, this has 2 plus 2 leaves, so it's 4 leaves, you will always have n leaves. So that's where I got n squared from. But if you look at this graph, it's actually blowing up only around this point where n is 12, and then it's kind of getting seriously bad after n is equal to 16 or 17. So I'm still figuring out, is there an implementation problem, or is this calculation of n squared wrong?
Okay.
Is there anybody? Our next speaker is, and probably the last speaker in this row, Muhammad Wasim from Intel Labs. Over